Welcome again. Today we look at topic 2.3 for IB Biology, fatty acids. A triglyceride is a typical example. It is the product of a triol, an alcohol with 3 OH groups and 3 fatty acids, with 3 fatty acid groups forming 3 ester bonds to this glycerol. 3 water molecules are lost by a condensation reaction. Triglycerides are typically found in fat tissue and in sunflower seeds. Depending upon the nature of these fatty acids, we can get different types of fats. Fatty acids can be saturated, monounsaturated, or polyunsaturated. Unsaturation referring to the presence of a double bond between one carbon atom and another carbon atom. A monounsaturated fatty acid would contain one single double bond, and a polyunsaturated fatty acid would contain several such double bonds. When we consider these double bonds, however, we do not consider the C double bond O of the carboxylic acid group. In the past, the general belief was that saturated fatty acids were the primary reason for the development of plaque or blockages of arteries. When fat blocks an artery, it takes up space in the lumen, and this can affect the flow of blood, leading to a range of health problems. But more recent research suggests that among the unsaturated fatty acids, there are 2 kinds, the cis fatty acid and the trans fatty acid. The cis fatty acid arising when the 2 hydrogens of the double bond are on the same side, and the trans fatty acid occurring when the hydrogens are on opposite sides. And recent research suggests that cis unsaturated fatty acids are linked to good health, and trans unsaturated fatty acids are linked to blockages of arteries and veins, with saturated remaining at its original status, and no definitive evidence is available to show that it has a positive health effect or a negative health effect. It is very important for the student of IB Biology to search for this evidence and to make an evaluation based on the current evidence in this field. As clinical studies continue to be published and more data becomes available. A quick way, however, to assess one's health risk due to the intake of fats is to calculate your body mass index. But this in itself needs to be subjected to an evaluation. But what is it about the structure, the cis fatty acid versus the trans fatty acid, that could possibly lead to a situation where one has a positive effect on health and the other a negative effect? This is most likely due to the fact that the cis fatty acid has poor packing between one chain and another chain. This poor packing leads to weak intermolecular forces and then to a low melting point, meaning that it is more likely to exist in the solid form at body temperature. Whereas trans fatty acids have good packing and by extension stronger bonds between one chain and another chain. This stronger intermolecular force ultimately leads to a higher melting point, making trans fatty acids less likely to be associated with the development of blockages known as plaque.